he and I have been friends probably for 30 years, but haven't seen each other for about 25. So, which is one of the reasons I suggested him as a guest speaker here, in hopes that you'd bring him out so we could reconnect. And also because of the great work that he does. I'm sure you find it really interesting. So a little bit about his background. He was at Boston University between 1984 and 1988, working with Lynn Margulis as his advisor. He was a postdoc in the Center for Great Lakes Study, which is part of Great Lakes Studies, rather. He's part of UW Milwaukee, working with Ken Nielsen from 1988 to 1991. And then from 91 to 95, he was at Yale University School of Medicine, first as a postdoctoral associate, an associate research scientist, and then adjunct assistant professor of medicine. Then he ended up in industry for a number of years. And I believe it was about 2010 you were in California.
Yeah, that's right.
He's now at the California State University at Northridge as a professor of biology. And Dave and his lab have been working on is looking at Salmonella, anti-cancer target organism in patients that would decrease its pathogenesis and increase its ability to target and colonize tumors. But Dave is actually a mycologist at heart. He's the president of the local mycology society. He offers courses in mushroom identification. And if he's not in his lab, he's out actually in the woods looking for morels and chanterelles. So I guess without further ado, Dr. David Bermudes, and on behalf of the Microbiome Center, we appreciate you being here. And,
really interesting to me. So I'm partly going to tell you about a historical journey, and then I'll bring you all the way up to the present with what I'm doing.
Try clicking at your screen to grow on PowerPoint.
Yeah, there we go. So this is an interdisciplinary study, that, but I want to point out I'm a microbiologist, not an oncologist, even though I've been forced to learn a lot about oncology over the years. So many of you know that if you have a tumor that's localized, like this giant cell tumor in the distal ulna that's causing this bone to enlarge, that if you treat that, if it remains localized and you treat it with surgery, there's actually no further consequences of the cancer. It doesn't spread if you can remove it from the primary site before it spreads. And so it might be hard to throw a Frisbee, but the patient is fine. And you can contrast that with things like melanoma, where it starts as a normal mole, which many of us have, but it becomes changed or transformed and can start to migrate and ultimately spread throughout the body. And when it does that, it's highly lethal. So this is an example of metastatic melanoma. And these are the superficial skin tumors that occur, but it's the ones where it metastasizes to lung and brain that make it lethal. I don't have the clicker. So this is a diagram of that process of the primary tumor cells changing and becoming metastatic. So what it's showing, for example, is that there are cells that are breaking out of this basement membrane that contains the primary tumor. And one of the ways they do that is with matrix metalloproteases. They can break down, they're also known as collagenases, and they break down the collagen that is keeping them contained in that primary tumor. They also go from these nice, smooth little rounded edge cells to cells with philopodia on them. And that's referred to as the EMT transition, where they become motile, and that's part of their ability to get around. And what they do is they get into the bloodstream. So if that's the bloodstream, you have the endothelial cells, and they actually squeeze between the endothelial cells and get into the bloodstream. And that's referred to as intravasation. And then they travel to a distant site and they get back out of the capillaries, and that's referred to as extravasation. So that process is what leads to metastasis. And a lot of people have been saying that and describing that for a long time. It's known from many different angles. So this is Watson's book. I think this was like a 1988 edition. It's pretty old. But look what he's saying. It's almost the same thing. They're showing the cell, and what he's saying is that they have to change to become that way. And that the properties that they have, they become like white blood cells, like, for example, macrophages that have that same ability to get in and out of blood vessels. And so in 1992, I met this guy, John Pollock, and unfortunately he just passed away last year. And when it came out, he was just fascinated with it because it was proposing an alternative way for cancer cells to metastasize. And that is that to gain those properties of white blood cells, rather than the other way that we know, which is accumulation of mutations, dysregulation of gene systems that then get selected for those properties, that this paper was saying, well, melanoma may actually hybridize, create a gene fusion with the cancer cells and give them that hybrid property. And John actually spent the rest of his career working on that. His last paper was published in 21. And he actually did prove that at least some cancers are that way. They were able to do it by genetically tracing a cancer in a patient that had had a blood transfusion from another donor, where the donor was actually known. So what they proved was the cancer cells had the genetics of two different origins. And of course, another thing that's a hallmark of cancer is maybe 90% of them have go through a tetraploid stage and then become aneuploid, losing some of the genes. And that seems to greatly facilitate loss of viability by compensating with extra genes. And so at that time, I got interested in this, and we actually started playing around with dual labeling of antibodies. And just some of the things that we were learning that was macrophage-like characteristics is, again, the motility, the extravasation, proteases, and also, interestingly, phagocytosis. So cells like, John primarily studied melanoma. And cells like melanoma, there's no textbook that's going to tell you that melanomas are phagocytic. Likewise, things like MHC class 2 and toll-like receptors are generally thought of as, macrophage characteristics, although now they're becoming much more widely known to actually be on different cancer cells. And of all of these, the one that caught my eye was CR3, which is complement receptor 3, because complement receptor 3 is one of the ways that microorganisms get into cells. They use that as a receptor. When you go into a macrophage through CR3, you bypass the oxidative burst, and that's a survival mechanism. And so somewhere along the line in there, I had the idea, well, if they're macrophage-like, could they be susceptible to macrophage pathogens? And I was in an infectious disease section, and there were people working with all of these. So I had access to all of these organisms. The first one I ever tried was Leishmania. Leishmania did actually attach to cancer cells, but Leishmania is an incurable disease. So it's probably not a good vector to be developing. But one of the other ones was Salmonella. And so we started looking at Salmonella interaction with melanoma. Again, no textbook will tell you that melanocytes or melanoma are going to take up Salmonella, but this is a melanoma, and each one of these little dots is a Salmonella that's entered into it. It's just a DAPI stain. We were kind of surprised by that, and encouraged by that, and thought, well, maybe you could actually target tumors with Salmonella. I should mostly say John at the time, because we had this idea that the bacteria, if they're invasive, are going to be good at getting into cells. He actually selected for a hyper-invasive strain. And the serendipity is the /pure minus. And I'll show you what I mean in a second. So pure minus means purine biosynthesis minus. So if you take wild type, or this clone 72, which is right here, and you inject it into a mouse, and you look at an early time point, you will find the bacteria in the blood, smaller amounts in the tumor, and also in the liver, and similar proportions. And so I would call that dissemination. They pretty much go everywhere. And that's what Salmonella has the ability to do. Clone 72, not much different. But the interesting thing is that if you come back the next day and harvest the tumor, these are CFU assays. If you've taken microbiology, we're actually grinding tissue, plating serial dilutions, and counting colonies. So you come back the next day, and wild type has gone down, but clone 72 has gone down even more in the blood. In the tumor, there's a massive proliferation of the bacteria. We've gone from 10 to the 4 to 10 to the 9 in one day. And in the liver, they're going up for the wild type and staying about same for the clone 72. Now, if you calculate the tumor to liver ratio, which is more in the liver than there is in the tumor, it flips around more than 1,000-fold. And you have more than 1,000-fold more bacteria in the tumor than you have in other tissues. And also, I didn't put the dose up there, but the dose is 10 to the 6. And so now we have more bacteria in the system than we even started with. And there's an EM of the actual tumor, because we can see, this is B16 melanoma, because you can see melanosomes, we know we're looking at a tumor cell. And one of the first and surprising things that we saw was we didn't see a lot of bacteria in the tumor cells. There's one, and maybe there was another one I saw, but these are all the other gram-negative bacteria, and they're mostly on the interstitial space. So that was a little hint that maybe the concept of targeting by internalization wasn't going to be the only story. And I want to go back one and then just mention this, because, and for students, this is a really good lesson. When we first did this experiment, we wanted to believe that the better result, this improvement in clone 72, was due to the hyperinvasiveness. But what's known is that when you mutate bacteria and you make them oxytrophic for things like purine, these are adenine mutants, they become attenuated. They become less virulent. So you can look at this data and reinterpret it as, the biggest difference in the numbers is the dropping of the normal tissues relative to the other one and the expansion. So we started to believe that it's really the attenuation that helps foster this tumor targeting effect. Of course, we're getting it with wild type, but those mice only live for 3 days, 3 to 5 days. And I'll show you in a minute, these mice actually survive.
Question, if it's okay.
Yeah.
Is there a difference between the wild type and mutant on their intracellular invasion of the tumor, of the cells?
You mean in a EM?
Yeah, rather than being in the interstitial space, would, let's say, a wild type be more likely to be inside the cell?
I don't know. I think it's a great question.
Yeah.
Yeah. So much of this data was back at Yale, and I didn't have it all. We were collaborating with other people and working through dermatology. So I don't actually remember what the control picture looks like.
Yes.
But it's a great question. And I think similar thing will come up in a minute or two as well. This was one of the experiments I did, was that we had engineered the bacteria to have the herpes simplex thymidine kinase. And thymidine kinase can phosphorylate a nucleoside analog and become trapped in a localized manner. Can also activate drugs like ganciclovir and acyclovir. We actually showed that they could do that. And so what this is a radioactive version. So this is a tumor that's been excised, and they've been given salmonella with the thymidine kinase, and then we give them C14 FIAU, which is the nucleoside analog, and look for the accumulation. And so the interesting thing is, if you follow the accumulation, there's necrotic tissue is where they're being localized, and the more viable part of the tumor is where you find less of them. So this again is saying something slightly different than we first thought we were looking for, which is that they're going to a place that's different, that's got hypoxic region, includes dead cells, and dead cells, for example, release things like purines. So it becomes a food source for them. And this is also a region of the tumor that the immune system is relatively ineffective at reaching. So it's what's referred to as an immunoprivileged site. And so both of those things are probably contributing to this ability. And the reason that that's important and got a lot of notoriety is correlated with why we have such difficulty treating tumors. So here's a tumor. They have really irregular vasculature. When a tumor grows, it has to recruit new vasculature. And then they get this necrotic region in the middle. So when you give an anti-cancer drug, it goes through the bloodstream, and then not so good at getting through the tumor because its delivery mechanism is limited at that site. And what happens is that all the drug is efficiently delivered to the normal tissues with normal vascularization, and most of the drug can't get into the tumor and the necrotic region where viable tumor cells still reside. So you have what you already know, which is inability to treat the tumor and side effects because we're mostly treating the non-tumor tissue. So what makes it interesting is that this was the hard part to get to was the part that was being selectively colonized. And this was done by somebody else, and it's really shows you the progression of what happens when you initially inject and allow it to disseminate, and then you find it localized. So a short time after, so these are bacteria that are engineered with luciferase, and you can pick it up in one of these iVis cameras in a dark box. You can visualize it live, real time. And so you see that the bacteria have gone everywhere. A little bit later, you're starting to find them more localized in the two biggest places where bacteria go, the liver and the spleen. But then coming back later, we're primarily finding bacteria in the tumor. It's a tumor that's been implanted right there. Now, what I think is not happening is what seems to be happening, which is like they're migrating to the tumor. But I believe what's happening is they're being eliminated at all these other locations, and that they're preferentially growing. Because again, even if you take all this amount of bacteria and put it in here, this is only 10 to the 6, and this is 10 to the 9. So they have to be growing. And this was another experiment. That bioluminescence approach can't be used in humans. We don't have a giant box yet for humans, but that may come in the future. But PET imaging is one that can be done, and the same TK system can be done, and we were able to localize tumors based on that. So these bacteria may have some interesting potential as diagnostics in humans if this same phenomena can occur in humans. Well, I didn't mention it, but people were telling us we were crazy because we wanted to take sick people that were sick with cancer and inject them with a bacterium that's salmonella. So we didn't get a lot of love early on. But later on, people started coming around a little bit. For one thing, when we started, we didn't know that there was a long history of bacteria and cancer. So William Coley, more than 100 years ago, was using bacteria and bacterial extracts to inject into patients with soft tissue sarcoma and getting some pretty interesting results. And he's actually credited as the person that's the father of immunotherapy, because it was almost certainly eliciting an immune response. The tumors actually turned hot, and that's one of the current themes in oncology today, which is the checkpoint inhibitors literally can take cold tumors and make them immunologically inflamed and actually change the temperature. So we learned about Coley. We also learned about other earlier work that I won't go into. And then some stuff was popping up, while we were working on this, and people talking about spontaneous remissions associated with bacterial infections. So we, took this as a positive sign. And of course, 20 years later, we're having some interesting new results, or maybe 15 years later, because the first report, this was 2020, but the first reports were more like 2015, that people were finding a lot of bacteria in tumors. And in fact, they were finding a diverse microbiome in tumors. And not only that, but the biomes are different for different kinds of tumors. So it's tumor type specific. And even more recently, people have published that even the subtypes of breast cancer have their own specific microbiomes. Well, what I wonder and don't know and I'm interested in is whether any of that kind of transition that the tumor cells undergo to become metastatic, becoming phagocytotic, influences what microbiome they get. But that's something we don't know the answer to right now. Oh, and the other thing is there's more evidence right now that these microbiomes are more like drivers of cancer metastasis than regulators or suppressors. So unlike those infections with pathogens, the normal microbiome may be driving metastasis. And this is a recent paper where they treated with antibiotics and got lower amounts of metastasis. And antibiotics had no effect on primary tumors, only had an effect on metastatic tumors. All right, so back to salmonella. When you are a mouse with a tumor and you get salmonella, your tumors don't grow as large and you live longer than you would without the salmonella. And that's what this shows. So the control gets to a large size. They have a cutoff for where they get euthanized, be a little bit lower than it today than it was in this diagram. So these are essentially censored. And you give the salmonella at that same dose that I mentioned of 1 × 10⁶, and you can almost flatline the tumor, not quite. It will ultimately grow. So it's not a cure, and it's not even a regression. But this slide also illustrates what I mean about the therapeutic potential. Because if you need to take a Tylenol for headache, you're deciding whether you're going to take 1 tablet or 2. And if you have kids, you're deciding whether breaking it in half is about right. But you wouldn't take a Tylenol and break it into 10 pieces and expect to get almost the same activity. And you wouldn't expect to take one of those 10 pieces and break it into 10 again and still get quite a lot of activity. So there's something very different about salmonella, and it is the fact that it's replication competent. The whole dose doesn't need to get to the tumor if it's ultimately going to expand to 10⁹. And the other thing that we've learned, I'm not going to go through this in detail, is, and it's not that surprising, is that the bacteria are activating the immune system, and they're also regulating the tumor and even the tumor cells. So some of the examples are IDO, which is involved in the immunosuppression, involved in checkpoint inhibition. That actually goes down. CX43 is one of the connexins. It's actually upregulated, and they increase antigen presentation. So they're recruiting, and they wind up recruiting other cells. The bacteria themselves have LPS and flagella, so they're stimulating interferons. They're activating T cells. Macrophages are also getting activated. They're making things like tumor necrosis factor, and that has an antiagenic effect. And there are other things that people are still discovering that seem to be this like multifold mechanism of bacterial inhibition of tumor growth. Here is an example of what happens in a lung tumor, because B16, if you inject it in the tail vein, can metastasize to lung. So that's a normal lung. And that's the lung of a mouse with metastatic melanoma. So you can still see some normal tissue, but many, many black nodules. You also see some amelanotic nodules. That's something that seems to evolve right in the mouse. It's not that uncommon to have amelanotic ones. And then that's the treatment with the salmonella, suppressing the tumor growth pretty significantly. So gram-negative bacteria are highly inflammatory, and they cause what's known as septic shock. That leads to what's known as DIC, disseminated intravascular coagulation, which is lethal. And So again, our friends are saying, you know, this still isn't going to be safe. So literally people that were even my friends were saying, you're going to kill the first person that you inject this into. So we were aware that not all of the lipids in the normal lipid A are necessarily inflammatory. There's some that are more inflammatory, like Neisseria, and there's some that are less, like Bacteroides. And there was a paper published right around the time that we were concerned about that problem. And it turns out the gene that adds the myristyl group, if you knock it out, the lipid becomes non-pyrogenic. And so we immediately began doing that in Salmonella. And this was the result. So one thing is you can't use mice for this kind of example, this kind of test. Mice, although they make a ton of TNF-alpha, they're not sensitive to TNF-alpha. But other animals like pigs, these Sinclair swine, are extremely sensitive. So that one single mutation went from 100% death of the pigs in about 3 days to 100% survival. So extremely attenuating mutation just by deleting MSPD and satisfying what at least the apparent requirement is of suppressing the virulence aspects, especially septic shock. Now, I'm not 100% sure that you couldn't achieve that with a lower dose, but at the time, it was almost like a mandate that we do something along those lines. And this went into human clinical trials. So what this shows on this side is a dose escalation. So they made us start in humans at a dose that was actually 5 times lower than a safe dose for a mouse and progressively go up. And they were able to do biopsies and do a CFU assay. And you can see there were a lot of zeros early on. By the time we got up to around 3 times 10 to the 8, they actually started detecting bacteria in the tumors. One of the things I'd note that is that they had 2 different methods. FNA is fine needle aspirate, whereas excision is actually a surgical removal. And in this one patient, the excisional found the bacteria and the FNA didn't. And I think that's because the bacteria are not necessarily localized throughout the entire tumor. They may have smaller niches, and if you don't hit it with the needle, you don't get it. Later on, we had some additional ones, and some of them went all the way to what we had in mice. But overall, you know, this is what I call my scorecard. Like how do we do? You know, and how the mouse model stack up to humans, because it's humans that count. Many people have cured mice. So it's not surprising that you can get things to work in mice. Well, one thing is at least I got the acceptable toxicity. No one in the study died. About 1 out of 3 were colonized. So at the MTD. So that's less than mice. We have almost 100% colonization in mice and only about 30% in humans. And also the number of CFU was lower. We saw 1 with 10 to the 9, but more with more like 10 to the 5. Just checking the time. So what my goal became is, if the bacteria can target tumors, what can I do to improve? Oh, and there was, sorry, I guess subconsciously, I hate to say this, there was no anti-tumor activity. Yeah. The also thing I would point out, it was really skewed towards melanoma. And the reason for that was, this was done at the NCI, and the person there, Steve Rosenberg, was the chief of surgery, and he was studying immunotherapy in melanoma and renal patients. So these were the patients that failed his therapy. So one of the questions I get asked sometimes is, would you expect people to respond to salmonella therapy if that's mostly an immunotherapy and these people already failed immunotherapy? But it's speculation. Some people also ask what would happen in other patients, and there's just no answer at this time. So what can I do? What can I do to take these bacteria? They're there at least sometimes in the humans. They're not creating anti-tumor activity, at least that's what's documented. What can I do to make them better? You know, essentially push their way out and kill more cells and have something that you can recognize. So one of the things I got interested in, there's a group at the NCI that's been taking bacterial toxins like Pseudomonas exotoxin A and conjugating it to antibodies or other ligands and treating patients that have tumors that are overexpressing the receptor for that ligand as a therapy. And one of these got approved recently for hairy cell leukemia. It's targeting IL-13. So we engineered some that have TGF alpha ligand, which targets EGFR. We're able to show that the wild type toxin kills all the cells, but when you make that transition, it only kills the, effectively kills the EGFR positive cells. We also played around with some different forms. These are chimeric fusion proteins. We played around with some different forms. We're actually able to make one that was specific for cancer cells and stronger than the wild type. But when I tried this in a mouse model, I could detect of an EGFR positive cancer, I could detect no anti-tumor activity. And this is there's a little bit of scatter there, but I did all the tumor measurements. Every mouse in this study looked exactly the same. They looked exactly like the saline control. And I was pretty disappointed by that. Started thinking of some of the reasons that could have occurred. And one thing is, so here's the TGF alpha. Wild type toxins, a lot of the proteins bacteria make are relatively resistant to proteolysis. But once you start adding things on, especially with linkers, you can start getting sensitivity to proteases. And so we hypothesized that maybe it's a protease-related problem, and had the idea of one way to solve that would be to make protease inhibitors against some of the proteases that are common in tumors. And among two of the most important ones are the metalloproteinases, and that's actually what you saw on that diagram. And another one is the serine proteases. So the first one we engineered is called sunflower trypsin inhibitor. It's a small little peptide. And we added it to this protein, YebF, which is an E. coli protein of unknown function that's used as a secretion carrier. You can actually buy it as a plasmid where you can clone in the stuff you want to get secreted. And so we generated that, and we had so we took some tumor cells, we had untreated control, we had some with just some culture supernatant, and then the toxin that we had made, and it killed all the cells, basically zero, this toxin. Then we treated this toxin with trypsin, and all the killing went away. But then we put this polycistronic one in, and the killing went back to 100%. Well, the part I'm leaving out is that didn't solve the problem in my EGFR positive cancers. But I found something serendipitously, which is that carrier protein, YebF, has an anti-tumor activity. So it's another good lesson, which is that the control is the best thing in the whole study. So you can see untreated, you can see, so VNP was the lipid A mutant one. It suppresses, but eventually they grow. The one with sunflower trypsin inhibitor was pretty good, but the YebF was actually slightly better. And that held true for the tumor volume and even the survival. Here's the control mouse, and you're going to have difficulty seeing that teeny little scar with the red arrow is where the tumor was. But the other thing that occurs is there's outgrowth from that region. So that's why this curve goes back up. But when you go in to look at the mice over this period, this really dramatic regression of the tumors. So what is YebF? Well, it turns out it's a really widely distributed family of proteins, and all of unknown function, with the exception of one that's colicin M. And colicin M is an antibacterial colicin. And what it does is it dephosphorylates the undecapryl phosphate and prevents that cell wall assembly process that occurs in bacteria. And so because it's doing that, it's a phosphatase. So we wondered if it could be a phosphatase. And I compared that with YebF. And I'm going to go back one. And D226 is one of the ones that's been mapped as potentially important. It's an aspartic acid. And so I compared them, and it did not have a homologous aspartic acid, but in a region of small similarity, there was a conserved aspartic acid dimer. So we wondered if this could be a phosphatase. So I actually just took E. coli and put in the YebF plasmid. And I had, you can get a YebF minus E. coli. There's culture supernatant. So here's the wild type E. coli. They have a YebF, and they give some phosphatase activity. When you delete YebF, it goes down, it doesn't go to zero, but then you add back the YebF plasmid and it goes back up. So there seemed to be some kind of association of YebF with phosphatase activity. So then I took some inhibitors of serine threonine phosphatases and tyrosine phosphatases. And I also tried heat treatment. And so here's so YebF, the VMP with YebF had a lot of activity. And if you treat it with a serine protease inhibitor, there's nothing. If you treat it with a tyrosine phosphatase inhibitor, there's pretty good inhibition. So we had the idea that it might be a tyrosine phosphatase. So one experiment I did, this is pretty recent, like very current. One experiment I did was take a culture supernatant and to see what would happen if I exposed it to tumor cells and look for the release of phosphate. So I did that both with an empty vector of my salmonella and one with the YebF plasmid. And in fact, more phosphate is released by the culture supernatant from the salmonella. And that is pretty much where I am right now. I've got a potential collaborator for actually identifying what proteins get dephosphorylated using mass spec. So that's really one of my near-term goals, to try and understand. And the other thing is, I feel like if I can understand what the target is, maybe someone else already has a therapy that interferes with that target. Perhaps it's a kinase, because the kinases are the ones that do the phosphorylation. And kinase inhibitors are one of the approaches to cancer therapy that's been pretty successful over the last 10 or 15 years. So it's possible someone already has a kinase that puts that phosphate on, and mine has the enzyme that takes it off. And what's interesting to me about that is, if they have a monotherapy with that kinase, they may also have a combination therapy that works. And it could actually tell me what therapy, because remember, my tumor is growing out again. It's regressing, it actually shrinks, and it almost looks like you're going to get a cure, and then it starts coming back. So I'm looking for that thing to be able to combine with my salmonella therapy. So these are the future directions I think I just said. Also, extracellular tyrosine kinase phosphorylation wasn't known till about 2015 or 2014. The first one was discovered, but it turns out they are involved in cancer progression. And so it's possible that I'm hitting an extracellular target, but it's also possible that it's getting trafficked through the endocytic system and having its result on some other phosphatase. But that's what I'm hoping to learn in the future. So I'm at Cal State Northridge. These are the people that contributed. Many of these, Jamie, David, and Lena Vincent were all my master's students. We only go through the master's program at Cal State Northridge. And then Michael Summers is a professor emeritus, but he's great at protein purification. And so he's going to be working on this with me. He also already did the partial purification of YebF. And the other people, Jacqueline, Andrew, Tony, and Jessica, are the ones helping me with the mouse experiments. And they mostly help with weighing the mice and doing the implants, but I do all the measurements myself. It's one of those things that needs to be done by a single person. There's variation in how you use the calipers, and it's important to do it that way. So thank you very much.
All right, we traditionally do questions from